biology and the biochemistry has been worked out here. Now, much of that, for understandable reasons, has been focused on the hematopoietic system. There's much less detail available on non-hematopoietic cells, and Tommy's going to, our speaker, Tommy Liu, is going to address that today in the context of chondrocytes and cartilage. Tommy comes to the Institute from, well, China, I guess, initially via New Zealand. He did a bachelor's degree in pharmacology at the University of Otago, followed by a master's in biomedical science at the at Victoria University in Wellington. He's won a swag of best student presentation prizes throughout this PhD. I think the one that he's most proud of is the 3-minute thesis presentation run several years ago by the University of Melbourne, and so Tommy was able to summarise in a kind of wrapper version of his PhD in 3 minutes, and he won the People's Choice Award. So he's not going to do that today. He has a little bit more time, and we look forward to hearing the story. Thanks, Tommy.
Well, thanks, Ian, for that introduction. My name is Tommy, and today I shall be presenting findings from my PhD, which is focused on the role of SOCS3 in chondrocytes during development and in inflammatory arthritis. Rheumatoid arthritis is a systemic autoimmune disease that affects about 1% of the population. It typically manifests as chronic inflammation that affects numerous joints within the body. Features of joint inflammation include swelling, pain, and stiffness within the joints that can even eventually lead to joint remodeling. Now, a major feature of rheumatoid arthritis is the progressive destruction of cartilage, and it is one of the major components of the deformity and progressive disability that occurs in patients. Now, this process is irreversible, and other than partial and full joint replacement, arthritis is not yet a curable disease. Cartilage, which lines the connecting ends of bones, is unique in its ability to absorb compression while also resisting shear forces during joint articulation. Now, chondrocytes are the resident cell type in cartilage and are responsible for producing the proteins that make up the extracellular matrix. Two major proteins are type 2 collagen and proteoglycan. Type 2 collagen gives cartilage the tensile strength it requires to resist shear forces. It also provides a docking scaffold for other proteins such as proteoglycan. Proteoglycan forms an aggregate with the hyaluronic acid backbone and becomes trapped within the scaffold. And this is a highly charged molecule that is able to recruit and retain water molecules, allowing this tissue to swell, giving its ability to absorb compression. Now, chondrocytes are not only play a key role in maintaining articular cartilage, they also are critical towards joint inflammation, skeletal development. Now, the growth plates are centers of bone growth at the ends of long bones, and chondrocytes play a critical role here by laying down a cartilage template for bone formation. Now, here, cartilage can be separated into different zones depending on their stage of differentiation and function. Chondrocytes at the reserve zone are a subset of stem cell-like cells that replenish the proliferative zone. In the proliferative zone, chondrocytes divide along the long axes of the bone in regular columns, and this is considered the active site of replication and is crucial to bone elongation. From the pre-hypertrophic to hypertrophic zones, chondrocytes cease proliferating and begin secreting factors that drive bone mineralization. In the hypertrophic zones, hematopoietic cells and osteoprogenitors invade and occupy cavities left by apoptosed chondrocytes, eventually forming the bone marrow. In the ossification zone, osteoprogenitors differentiate into osteoblasts and deposit bone matrix on the cartilage tuberculi. Now, currently, there are no therapies that can regenerate cartilage. However, there are agents, both biological and conventional, that can alleviate some of the other symptoms of rheumatoid arthritis. Non-steroidal anti-inflammatory drugs, such as ibuprofen, are a temporary form of relief for pain and swelling, However, this does not halt the progressive destruction of your joint. Disease-modifying anti-rheumatic drugs, such as methotrexate, have been shown to delay cartilage deterioration. However, there's limitations with this approach, including toxicities and non-specific mechanisms of action. Now, biological therapies that target specific cytokines have really revolutionized treatment for patients. Now, TNF inhibitors, such as etanercept, has been shown to decrease disease activity while slowing down cartilage degradation. However, a significant number of patients still do not respond to this treatment, and hence the requirement to discover other targets other than TNF. Now, the anti-interleukin-6 receptor antibody tocilizumab has been shown great promise in the clinic and in clinical trials for not only rheumatoid arthritis, but other inflammatory diseases. And it's really been due to the success that other interleukin-6 family members have gained interest as mediators of inflammation. So where are all these cytokines coming from? What cell type or combination of cell types is initiating and driving joint inflammation? Well, in inflammatory arthritis, most attention has been given to the cells of the synovial tissue, such as synovial fibroblasts, macrophages, and osteoclasts, and the cytokines that they produce, such as interleukin-1 and TNF. And like I said, this has led to the development of successful biological therapeutics that antagonize these cytokines. Much less attention has been given to the role of chondrocytes and their potential contribution towards joint inflammation. While these cells have a recognized role in producing and maintaining cartilage, how this is activated and whether these cells can respond to interleukin-6 and its family cytokines is not well understood. Now, in order to elicit a biological response, each GP130 cytokine utilizes the JAK-STAT signaling pathway. Binding of the cytokine to this GP130 subunit with the receptor results in the activation of JAK kinases. Now, these phosphorylate tyrosine residues on the intracellular side of the receptor, leading to the recruitment and activation of STAT proteins. STATs dimerize, translocate into the nucleus where they can induce gene transcription. Now, there are parallel pathways that can also be activated, and this is done through the SHP2 protein that binds to tyrosine residue 757 and the activation of MAP kinase and AKT pathways. Now, binding of the cytokine to the receptor itself isn't as easy as it looks. This is because the ligand binds to the GP130 subunit with low affinity, and it is only in the presence of its receptor, non-signaling receptor, can signal transduction be induced. Now, the receptor for interleukin-6 exists in a membrane-bound and soluble form and has been extensively studied. Soluble IL-6 receptor is able to amplify the effects of interleukin-6 and also allow it to activate cells that don't normally express the membrane-bound interleukin-6 receptor. Interleukin-11 has been shown to interact with its own non-signaling receptor. Oncostatin M, at least in mouse bone cells, has been shown to signal through both the osmo receptor as well as the LIF receptor. LIF, cardiotrophin-1, cardiotrophin-like cytokine, and ciliary neurotrophic factor all have been shown to signal through the LIF receptor. Now, the regulation of the JAK-STAT signaling pathway, both by duration and in magnitude, appears to be critical in limiting joint inflammation. Now, this has been shown with the success of the JAK inhibitor tofacitinib, both in clinical trials for numerous inflammatory diseases. Now, the endogenous regulator of the JAK-STAT signaling pathway is SOCS3. SOCS3 belongs to a family of 8 members, each with an N-terminal domain that is variable in length and function, an ASH2 domain that allows it to bind to phosphotyrosine residues, and a SOCS box. SOCS1 and SOCS3 are unique in that they also have a kinase inhibitory region. SOCS2 and cis are able to competitively bind to phosphotyrosine residues and prevent STAT binding. SOCS3 binds with high affinity to tyrosine residue 757 to prevent SHP2 protein binding. SOCS1 and SOCS3 can simultaneously bind to GP130 subunit and JAK kinase, and through its kinase inhibitory region, block substrate access. Finally, SOCS3 can recruit components of an E3 ubiquitin ligase complex and polybutyrinate JAK and target it for proteasomal degradation. Now, the global SOCS3 knockout mouse is embryonically lethal. This has been shown to be due to excessive lift signaling. This problem is overcome with the conditionally knocking out of SOCS3 using the LOX-3 system. Now, the majority of studies looking at SOCS3 has been conducted in hematopoietic cells, but we wanted to investigate whether SOCS3 has a regulatory role in chondrocytes. So we hypothesized that deleting SOCS3 in chondrocytes would increase their responses to GP130 cytokines, increase signal transduction, exacerbate any inflammatory responses they may have, therefore highlighting their role in disease. Now, in these mice, Cre recombinase is driven by the type 2 collagen promoter COL2A1, and SOCS3 is flanked by LOX-3 sites. So what this means is that in chondrocytes, Cre recombinase is active, SOCS3 is deleted, and in other tissues, Cre recombinase is inactive, SOCS3 is left intact. Before I wanted to conduct any in vitro or in vivo experiments, I first needed to confirm that Cre recombinase was expressed in the cartilage of our conditional knockout mouse, that SOCS3 was deleted, and investigate what are the consequences of this on JAK-STAT signaling. In order to answer that first question, I crossed our conditional knockout mouse with a ROSA 26 reporter mouse, and I hypothesized that in tissues with no Cre recombinase expression, LAG-Z gene is not expressed. And in cartilage, in the presence of Cre recombinase, the stop promoter is deleted, LAG-Z is expressed, which can be detected using X-scale staining. Now, by performing X-scale staining in littermate and SOCS3 deleted mice, I observed that there was no positive staining in the littermate controls, that there was positive staining in our SOCS3 deleted cartilage, and I used a ROSA 26 mouse that constitutively expressed LAG-Z as a positive control. Now, having shown that Cre recombinase is expressed in the cartilage of our conditional knockout mouse, does this lead to SOCS3 deletion? Well, in order to examine this, I needed a method to harvest and culture primary chondrocytes. In order to do this, I take the knee joints of 5 to 6-day-old mice, and under the microscope, I dissect the knee joints in order to obtain 2 pieces of epiphyseal cartilage. Now, these 2 pieces give you around about 250,000 cells per knee joint. And you can digest these enzymatically overnight and culture the chondrocytes in monolayers. So I took primary chondrocytes from wild-type, SOCS3 littermate, and SOCS3 deleted mice, stimulated them with interleukin-6, with its interleukin-6 receptor, and looked for SOCS3 mRNA expression. I used type 2 collagen and type 1 collagen as indicators of chondrocyte purity. This is because chondrocytes only express type 2 collagen, fibroblasts express type 1 collagen, and the major problem with many studies out there is the tendency of chondrocytes to de-differentiate into fibroblasts-like cells after prolonged culture. And you can actually demonstrate this with the expression of both type 2 and type 1 collagen. And for this experiment, I used a bone marrow stimulated with GCSF as a positive control. And what I found was SOCS3 was indeed upregulated in wild-type and littermate chondrocytes, and that it was absent in our SOCS3 deleted cells. Now, to examine the biochemical consequences of SOCS3 deletion, I performed intracellular staining for phosphostat proteins and analyzed the chondrocytes by flow cytometry. Now, for these experiments, I stimulated littermate chondrocytes in red, SOCS3 deleted chondrocytes in blue for 30 minutes, 1 hour, and 2 hours with GP130 cytokines, and analyzed the detected by phosphostat3 by flow cytometry. So activated STAT3 was detected 30 minutes after stimulation and began to return back to baseline levels 1 hour after stimulation, but was in our littermate controls, but was still elevated in the conditional knockout cells. Now, the differences were most evident 2 hours after stimulation when phosphostat3 was still detected in our conditional knockout chondrocytes. Now, what you may notice is that oncostatin M appears to be a potent mediator of phosphostat3 activation in chondrocytes. Now, interestingly, interleukin-6 stimulation by itself induced minimal STAT3 activation, and only in the presence of its soluble IL-6 receptor can these levels of activation be detected. So what this indicates is that chondrocytes would express low levels of the membrane-bound receptor and interleukin-6 requires its soluble IL-6 receptor, or trans-signaling, in order to mediate its responses. STAT3 isn't the only signal transducer downstream of GP130. STAT1 is the other. And so I looked at the kinetics of STAT1 and observed similar kinetics. I saw that STAT1 was activated in littermate chondrocytes 30 minutes after stimulation, and again, that its signaling was prolonged in our SOCS3 deleted chondrocytes. Again, what you may notice is that there are different kinetics of activation for STAT1 and STAT3 in chondrocytes by GP130 cytokines, even though they signal through the same receptor. And this is something of interest that we definitely want to follow up. And one possible reason for this could be receptor expression. And so I analyzed the expression of GP130 cytokine receptors in wild-type chondrocytes and looked at it by flow cytometry. So for this experiment, I stained chondrocytes with biotinylated antibodies for GP130, oncostatin M, interleukin-6, IL-11, and LIF, and came in with a secondary strep-evident PE antibody. Using the QuantiBrite bead system, I calculated the number of antibodies bound per cell using the MFI, which is an estimate of the number of membrane-bound receptors. What I found was that GP130 subunit is expressed at high levels, and interleukin and OSM and LIF receptors were expressed at high levels relative to interleukin-6 and interleukin-11. So what this result indicated was that oncostatin M can potently induce phospho-STAT3 and STAT1 activation by itself, and that interleukin-6 requires its soluble receptor in order to mediate its responses. So it is important to note here that I found that there was no difference in receptor expression between SOCS3 deleted and littermate chondrocytes, and that I admit there are limitations with this particular technique, and so we are planning to conduct ligand binding assays in order to support these findings. So summarizing that particular section, I confirmed that Cre recombinase is expressed in the cartilage of our SOCS3 deleted mice, and that SOCS3 deletion led to prolonged STAT1 and STAT3 signaling in response to GP130 cytokines. So when you generate a novel knockout mouse, the first thing people ask is whether there are any developmental defects, and it's especially true when chondrocytes play such a vital role in skeletal development. And to answer this question, we conducted an aging cohort of our conditional knockout mouse from 1 week to about 1.5 years old. The first thing I noticed was that the pups at 6 days old in the conditional in our SOCS3 deleted pups were smaller in size compared to the littermate controls, and that there was a consistently reduced total body weight in our conditional knockout mouse compared to the controls throughout their lives, and that this was consistent between male and female mice. It's important to note here that there were no differences in soft tissue weights, and so it suggested defects within the skeleton. So in order to examine this in more detail, I compared the femurs of SOCS3 deleted and littermate mice. And what I found was that indeed at 6 weeks and 12 weeks, SOCS3 deleted mice had shorter long bones compared to their littermate controls, and that this was consistent between male and female mice. So in order to examine what could be contributing to this developmental defect, I focused on the chondrocytes at the growth plate. Now, just to remind you guys, growth plate is the centers of bone elongation, and the chondrocytes at the proliferative zone play a crucial role in bone elongation. Now, in order to examine whether SOCS3 deletion had any effect on the proliferation of growth plate chondrocytes, I conducted Ki-67 staining, which is a cellular marker for proliferation in 6-day-old knee joints. What I found was that there appeared to be reduced Ki-67 staining in the growth plate of our conditional knockout mouse relative to controls, and that when we counted it using the metamorph system, even though there was no difference in the total number of cells, there was reduced Ki-67 positive staining. So what this indicated was that at this early time point, at least, decreased proliferation of growth plate chondrocytes could have led to the reduced long bone length in our adult SOCS3 deleted mice. So why would SOCS3 deletion in chondrocytes result in defects in growth plate chondrocytes, or growth plate proliferation? Well, GP130 cytokines have been demonstrated to play a role in bone growth. In particular, IL-6 transgenic mice that overexpress interleukin-6 have impaired skeletal development. Now, these mice are smaller than their wild-type controls and also develop shorter limbs. And this paper in particular suggested that it could have been due to abnormalities within the growth plate, although they only measured they looked at, in particular, the hypertrophic chondrocyte zone instead of the proliferative zone, which is directly above. So one potential explanation for why our conditional knockout mouse was smaller than their littermates could be prolonged GP130 cytokine signaling. However, there are other pathways that can also regulate bone growth, and one in particular that I am interested in is the fibroblast growth factor receptor 3 signaling pathway. Now, during normal skeletal development, FGFR3 regulates growth plate chondrocyte biology by inhibiting proliferation and also the differentiation into hypertrophic chondrocytes. Now, in humans, a mutation in FGFR3 results in overactivation and contributes to the major forms of dwarfism in humans. In mice, when you overactivate FGFR3, the transgenic mice are smaller than the wild-type controls. And when you delete FGFR3, they develop longer limbs. So there is some evidence to suggest that SOCS3 can be upregulated after FGFR3 signaling. However, this hasn't been examined in detail, and it is something that we want to look at and follow up in the future. So to summarize the developmental aspect, SOCS3 deleted mice are smaller than their littermate controls and have reduced long bone length. And I found that SOCS3 deletion in chondrocytes resulted in reduced proliferation at the growth plate. Now turning to the focus of my project, which has been on inflammatory arthritis. The first question we wanted to ask was what were the consequences of prolonged JAK-STAT signaling with regards to cartilage degradation. Just to go back and remind you guys, chondrocytes are produce and maintain the cartilage extracellular matrix, and that proteoglycan and type 2 collagen are two major signaling extracellular matrix proteins. Now, cleavage of these two major proteins leads to destabilization of the extracellular matrix and is the major feature of cartilage degradation. And therefore, there's really a lot of interest in how these cytokines mediate this process and how we can intervene in order to improve cartilage health. Two major families that are involved in cartilage degradation is the ADAMTS family and the MMP family. So ADAMTS are able to cleave the protein backbone of proteoglycan. ADAMTS4 and 5 are particularly efficient at doing this and have been shown to be induced by inflammatory cytokines. MMPs can cleave extracellular matrix and non-extracellular matrix proteins. And MMP1 and 13 are major enzymes involved in cleaving type 2 collagen. And MMPs have been shown to be induced by inflammatory cytokines. A less well-studied enzyme family in terms of cartilage biology is the cathepsin family. Now, members L, B, and K have been shown to be expressed in joint cells. Their function varies from cartilage degradation to bone erosion, and they have been shown to be upregulated in the synovial fluid and serum of RA patients. So in order to investigate whether cartilage degradation in our conditional knockout mice, I needed a method I needed to use cartilage organ culture systems. So the femoral head is the cartilage cap at the end of the femur neck, and with gentle manipulation, you can remove this, and it's a pure source of cartilage in 2 to 3 week old mice. So I took the cartilage explants, stimulated them with GP130 cytokines, and stained them with toluidine blue, which is a dye for proteoglycan. And what I found in littermate controls is that GP130 cytokines, to varying degrees, can induce proteoglycan loss, and that this was increased in the absence of SOCS3 regulation. Now, you can actually quantify this proteoglycan release using the 19-DMMB blue dye binding assay. And what I found was that in littermate controls, each GP130 cytokine can induce proteoglycan cleavage, and that in the absence of SOCS3, this was markedly increased. So how are chondrocytes driving cartilage degradation? What are the things that they could be potentially producing to mediate this process? Well, in order to examine this, I looked at the expression of ADAMTS4 and 5 in primary chondrocytes by qPCR. And this experiment stimulated littermate control and SOCS3-deleted chondrocytes with GP130 cytokines. And I found that indeed aggreganases 4 and 5 were upregulated in littermate control chondrocytes, and that this was significantly increased in the absence of SOCS3 regulation. Proteoglycan cleavage is one part of cartilage degradation. The other is type 2 collagen degradation. And so I also looked at the expression of MMP13 and 1, and I found that MMP13 in particular was upregulated potently in littermate control chondrocytes, and that this was again exacerbated in the absence of SOCS3 regulation. For MMP1, it appeared that in the absence of SOCS3, oncostatin M was the only GP130 cytokine that induced its expression. In terms of the cathepsin family members, 2 of the 3 were also upregulated in littermate controls and were also increased in the absence of SOCS3 deletion. So what this part indicated is that SOCS3 deletion exacerbated cartilage degradation in response to GP130 cytokines, and that SOCS3 really acts to limit the expression of aggreganases, metalloproteinases, cathepsins in chondrocytes during inflammatory arthritis. Having shown the regulatory role of SOCS3 in terms of cartilage destruction, I went to in vivo models in order to investigate whether these cells also played a role in the inflammatory aspect of arthritis. Now, the common readout for these in vivo models is histological scoring, and I scored disease based on 5 parameters. Pannus tissue is the formation of a hyperplastic flap of tissue over the bone and cartilage junction. Exudate is the presence and number of inflammatory cells within the normally clear synovial space. Bone erosion is a demineralization of bone. Synovitis is a thickening of the normally thin synovial lining by activated synovial cells and infiltrating inflammatory cells. Once again, cartilage degradation was graded upon the loss of toluidine blue staining. The first thing I wanted to examine is what is the contribution of each GP130 cytokine towards inflammatory arthritis. To do this, I took each cytokine and injected intra-articularly into the knee joint on 3 consecutive days, and at the height of disease, I took the knee joints for histological analysis. Now, saline was injected into the contralateral knee and acted as a negative control. What I found was that each GP130 cytokine can, to variable levels, induce joint inflammation and cartilage degradation. And that in the absence of SOCS3, this was significantly exacerbated. Interestingly, oncostatin M and interleukin-6 appeared to be potent inducers of inflammatory arthritis relative to interleukin-11 and LIF. Now, the KB-Bion serum transfer model is a model of acute inflammatory arthritis. It involves the injection of KB-Bion serum into the peritoneum and analyzing the ankle histology at the height of disease. Now, mechanistically, it involves the deposition of anti-glucose 6-phosphate isomerase antibodies directly onto the surface of cartilage and the activation of complement in Fc-dependent pathways. Now, this model is also dependent on inflammatory cytokines. Here, I've got some histology slides of saline, SOCS3 littermate, and SOCS3 deleted mice, H&E staining up on the top, and toluidine blue staining at the bottom. What I found was that in SOCS3 deleted mice, there was an intense increase in the destructive parameters of joint disease. In particular, exudate, synovitis, bone erosion, and cartilage degradation. And when you compared the histological scores, there was an increase in synovitis, cartilage degradation, and bone in our SOCS3 deleted mice compared to the littermate controls. Finally, the MBSA-IL1 induced arthritis model is a T-cell dependent model that, of acute inflammatory arthritis. It involves the injection of methylated bovine serum albumin into the knee joint on day 1, and followed by 3 daily injections of interleukin-1 into the foot pad, and the knee joints were harvested on day 7 at the height of disease. Now, the benefits of this particular model is that it's dependent on cytokines that signals through the JAK-STAT signaling pathway, and also because of its rapid onset, resolution, and short time course. Now, parameters of disease manifest with the classical signs of joint inflammation with exudate, synovitis, as well as cartilage degradation, as shown in the littermate controls. In our SOCS3 deleted mice, there was a marked increase in synovitis as well as cartilage degradation. Now, for this experiment, I also wanted to examine the contribution of chondrocytes compared to hematopoietic cells in terms of inflammatory disease. And so I also ran the SOCS3 VAV mice, which has SOCS3 deleted in hematopoietic cells and also endothelial cells. And what I found was that SOCS3 deletion in chondrocytes had as much a contribution towards inflammatory arthritis as the hematopoietic cells in this particular model. And so when I compared the histological scores, that's what I found, that there was an exacerbation of disease compared to the littermate controls, and that SOCS3 deleted mice had as much effect on inflammatory arthritis as the SOCS3 VAV-CRE mice. So how are chondrocytes driving joint inflammation? In order to answer this question, I went back to in vitro experiments and looked at the production of pro-inflammatory cytokines within the conditioned media. So for these experiments, I stimulated chondrocytes from littermate control and deleted mice with GP130 cytokines and found that SOCS3 deleted chondrocytes in particular produce large amounts of pro-inflammatory cytokines such as interleukin-1, interleukin-6, and GCSF, as well as pro-inflammatory chemokines such as CXCL1, the chemoattractant for neutrophils, and CCL2, the chemoattractant for monocytes. So what these experiments implicated is that chondrocytes during inflammatory arthritis can produce cytokines that activate other synovial cells and also recruit inflammatory cells into the local environment through the production of chemokines. Now, in our in vivo models, I also observed extensive bone erosion. Now, this raised the intriguing question of whether chondrocytes can also mediate bone erosion during inflammatory arthritis. To examine this, I performed micro-CT imaging of saline and arthritic knee joints in order to analyze the extent of bone erosion. Here are some representative 3D reconstructed models of knee joints. And what you can see is that there is extensive erosion of cortical bone in the tibial and in the femur of our SOCS3 deleted arthritic knee joints compared to the littermate controls. Now, if you use the colored scale here on the right, you also would see that there's extensive demineralization of bone in the tibial epiphysis in our SOCS3 deleted mice compared to the littermate controls. So how are chondrocytes mediating bone erosion? Well, the RANKL receptor activator of NF-kappa B and its ligand RANKL mediate osteoclast differentiation. Now, the production of RANKL by activated T cells in osteoblasts leads to the differentiation of progenitors into osteoclasts. And Osteoprotegerin acts as a decoy receptor for RANKL and is an endogenous negative regulator of the system. So in order to investigate whether chondrocytes produce RANKL, I performed immunohistochemistry staining of arthritic and saline knee joints during inflammatory arthritis. What I found in the saline controls was that there was RANKL staining in the bones of the knee joints, and that in the arthritic knee joints, in particular the articulate cartilage, there was positive staining of RANKL. In terms of OPG, I saw staining in both saline and arthritic knee joints, and it was difficult to determine whether there was an increase in the intensity of staining. In order to confirm this finding, I looked at the expression of RANKL and OPG in stimulated primary chondrocytes by qPCR. And what I found was that RANKL was upregulated in the littermate chondrocytes, and that this was markedly increased in the absence of SOCS3, although there was no difference in the expression of OPG. So the ratio between RANKL and OPG is a strong indicator of osteoclastogenesis, and increased osteoclast numbers drive a bone erosion. And so when I compared their expression, I found that oncostatin M and interleukin-6 were particularly potent at elevating the RANKL to OPG ratio. So what these experiments indicated was that chondrocytes could be potentially mediating osteoclast differentiation, contributing to the bone erosion that I was seeing in the in vivo models of inflammatory arthritis. Now, as a summary, oncostatin M appears to be a potent mediator of chondrocyte responses out of the GP130 cytokine family. Interleukin-6 appears to require its trans-signaling pathway in order to mediate its responses. SOCS3 is a critical regulator of GP130 cytokine signaling in chondrocytes. That when you delete SOCS3 in chondrocytes, it exacerbates joint inflammation and cartilage degradation in vivo. SOCS3 normally acts to limit chondrocyte expression of agrokinases, MMPs, cathepsins, as well as RANKL during inflammatory arthritis, and that chondrocytes are indeed active participants in joint inflammation through the production of pro-inflammatory cytokines and chemokines. Now, in the last 5 slides, I want to go through some ongoing work in trying to understand what makes oncostatin M such a potent mediator of chondrocyte responses. And to answer this question, we looked at the transcriptional profiling of chondrocytes stimulated with GP130 cytokines at the 4-hour time point. So this Venn diagram illustrates the number of up or down regulated genes in chondrocytes compared to unstimulated controls. So what you may notice is that the shared and unique changes in gene expression differed between each GP130 cytokine. In particular, oncostatin M induced the greatest number of unique and shared changes in gene expression out of the GP130 cytokine family. Now, in order to compare the magnitude of change between oncostatin M and the other GP130 cytokines, we generated scatter plots. So genes induced by interleukin-6, interleukin-11, and LIF are indicated on the y-axis. Oncostatin M induced genes are on the x-axis. Genes lying on the 0 point was not affected by stimulation. Genes lying on the 1-to-1 line demonstrated a similar response between oncostatin M and the compared cytokine. So what this analysis indicated was that the gene expression changes induced by oncostatin M highly correlated with those induced by interleukin-6 and differed most from those induced by interleukin-11 and LIF. However, if you look carefully, the oncostatin M-induced changes were more potent compared to interleukin-6, and that these changes were more evident when you compared them with interleukin-11 and LIF. So in order to determine what particular molecular pathways were selectively enhanced by oncostatin M, we performed gene set analysis that was developed here by Gordon Smythe. In order to perform roast analysis, a list of genes that is relevant to a biological process, such as positive regulators of inflammatory responses, is selected. Roast analysis takes into account the proportion of genes that within this list that responded to stimulation. This is indicated by the active proportion. Roast analysis takes into account the proportion of genes here that was upregulated, that was induced by oncostatin M or the other cytokine, for example, interleukin-11. It also takes into account the magnitude of change, whether it was large or small. And all of these factors goes into calculating the significance of the change. And so we analyzed gene set associated with various biological processes. And today, one pathway that I'll present today is the genes that positively regulate the inflammatory response. So the active proportion, just to remind you guys, is the proportion of genes within that list that was induced. And what this experiment indicated was that oncostatin M and interleukin-6 were similar, more similar compared to oncostatin M and interleukin-11 or oncostatin M and LIF. However, the genes within this gene set were induced more potently by oncostatin M compared to the other cytokines. So putting it all together, what does this mean? Well, it indicates at least that oncostatin M induces a potent inflammatory signature in chondrocytes compared to the other family members, which corresponds with the ability for oncostatin M to induce cytokine and chemokine production in vitro, as well as the pronounced joint inflammation and cartilage degradation I observed in vivo. Finally, future directions. While anti-interleukin-6 therapies have been successful in the clinic, many patients still do not respond to treatment. Therefore, targeting OSM or the OSM receptor could be a viable option in addition to targeting interleukin-6. Now, in mice, this has been shown with the neutralizing antibodies against oncostatin M and how it would reduce the severity of collagen-induced arthritis. Can SOCS3 be used as a therapeutic? Adding exogenous proteins has faced problems with delivery. However, I think that furthering our understanding of how SOCS3 is degraded, whether by proteasomal degradation or some other pathway, could allow us to prolong the half-life of SOCS3 in order to use its potentially anti-inflammatory properties. Finally, what I've presented here in my talk today is models of inflammatory arthritis. Now, a major form of arthritis is osteoarthritis. And OA is conventionally believed to be non-inflammatory, and chondrocytes play a large role, is believed to play a large role during disease. And therefore, it is definitely interesting to look at the role of SOCS3 in chondrocytes during models of OA. Now, all that's left is to thank some people. First of all, I'd like to thank my parents. They've been fantastic, supportive, couldn't have done it without them. In terms of people at WeHi here, Ian, Kate, and Ben have been fantastic supervisors and mentors. Ian, in particular, has really given me the chance to work independently, follow up things that I wanted to do. And also, he was very understanding after I ran that very expensive microarray. Kate is a terrific supervisor. She is an expert in cartilage, a terrific mentor, and also an excellent cook. And she should definitely go on to MasterChef or My Kitchen Rules. Ben Croker is a fantastic scientist. You cannot help but leave his office or a Skype chat feeling motivated and excited about your project and ready to face that 14-hour experiment that he so easily suggested. The rest of the Reed Lab has been fantastic in their technical assistance, their suggestions, their discussions, and really providing a shoulder to cry on in the last 4 years. Warren for being on my committee and allowing me to use the SOCS3 VAV3 mouse. The rest of the Reed Lab that I haven't mentioned. The Sandra Nicholson's lab has been fantastic in everything SOCS related in terms of molecular biology. Gordon and Cynthia has been so patient with me and explaining over and over again the microarray data. And thank you for doing so much work and helping me along in that aspect. Bilal's been fantastic. And histology and also imaging has been fantastic in processing samples and helping me along with the histology and immunofluorescence and immunohistochemistry. Fax Lab gets a mention. I couldn't have done it. Fax Lab already mentioned them. Couldn't have done it without the Mouse House girls. They have been fantastic, and they could have probably sat through the whole talk just to see their names here, and they are right there. The Murdoch Institute has shown me how to harvest chondrocytes, and finally, the St. Vincent's Institute is experts in anything bone or skeletal development related. Thank the APA, the Melbourne University, NHMRC for funding my talk, and happy to answer questions.
Nice talk. I got a little bit confused at the end with the analysis of gene expression. I wasn't quite sure I understood what you were trying to look for. to me, I guess the interesting comparison would have been if you'd have used a dose of those different cytokines that gave a similar output, and then asked whether there was a qualitatively different set of genes that were regulated. Sort of, it seemed to me that the things you were picking up was because you were hammering the cells with oncostatin M, but interleukin 6 was, or interleukin 11 and LIF were much weaker signals. So is it a qualitative difference that you're looking at, or a quantitative difference that you're looking at?
I think our analysis, I agree with what you're saying, that the experiment could be designed in another way, but the point of this particular analysis was to look at the gene expression changes in terms of, I guess, quantity, and that we also wanted to compare the changes in gene expression between littermate controls and SOCS3 deleted chondrocytes in order to see whether there are any changes in gene expression in the absence of SOCS3. So there was also that particular aspect of the microarray that I haven't presented here today that we were interested in look at.
Gordon, did you have any other comments on that one?
I'd say that, well, the gene expression, the expression response to the different cytokines was correlated, but for the non-OSM and interleukin 6, it was somewhat distinct. So there were things that you got in response. There were responses to the other cytokines that you didn't get from. So there were differences. It was not just a dose effect.
Well, but I guess the question there is if you dial down the dose of oncostatin M to the point where it is giving a similar magnitude to interleukin 6, whether they would all then look the same. In which case it's a quantitative response in a sense rather than a qualitative response.
The other data that you didn't mention was the phosphorylation 5 being induced in response to oncostatin M. So as a unique sort of response to oncostatin M.
Yeah, so there are definitely things that oncostatin M is doing that other GP130 cytokines it wasn't, and as Ian mentioned, STAT5 activation was one of the, it could have contributed to the difference in qualitative gene expression changes.
So that all those cytokines seem to have very nasty effects in the context of arthritis. What are the sort of physiological scenarios where they could have beneficial effects?
That's a really good question. Interleukin 6 in particular has been studied in terms of its catabolic roles in disease and also its anabolic roles. And in interleukin 6 knockout mice, they actually develop more severe disease in osteoarthritis models, suggesting that it has a protective or anabolic role, as you suggested. What I believe is the key to its mediating is its effects, its presence of soluble IL-6 receptor. In its absence, low levels of interleukin 6 stimulation could be anabolic and stimulate cartilage homeostasis, production of extracellular proteins, and that as soon as you get the presence of soluble interleukin 6, it reaches a threshold and it activates these other catabolic pathways as well.
I'm just wondering in terms of the model of what you think the SOCS3 is actually doing, and maybe this sort of follows on from that question. So you think it's limiting the response of chondrocytes so that they only produce a certain amount of output and not produce too much of it, so that then skews the response down one way rather than the other. So is that? I'm just trying to get to a model of what you think SOCS3's role really is.
I agree. I think what is happening physiologically is that SOCS3 limits and takes chondrocytes out as much as it can from their contribution towards inflammatory arthritis.
Is that what you mean, Chris?
Does that answer your question?
You're implying that...
Not really. I mean, the role of SOCS3 is not to stop rheumatoid arthritis occurring. Rather, it must have what is its real physiological role in terms of in chondrocytes? I mean, there must be is it just to limit cytokine production upon seeing these cytokines so that they do some things and not others? Or, you know, yeah, I'm just trying to get at what the physiological role is rather than what the phenotype is in the absence of that gene.
Yeah, I mean, that's a really good question. I don't think I've got an answer for you at this time.
I mean, I guess in terms of physiology, though, it's okay to think of a negative feedback mechanism for controlling cytokine responses for any cell. Another aspect that the microarray was the induction of the interferon response.
Yeah, so when you...
In the absence of SOCS3.
Yeah, so there was a change in the quality of the gene expression, change in the gene expression induced by GP130 cytokines in the absence of SOCS3 as well. So definitely it controls not only the quantity but the quality of the response.
Which has been described previously. So this is kind of yourself.
SOCS3 mutated. Is SOCS3 mutated in humans with rheumatoid arthritis or downregulated?
Well, that's really interesting. So what we've shown you is a model of cartilage deleting SOCS3 in chondrocytes. So there's some evidence to suggest that it's downregulated in osteoarthritic chondrocytes. However, those findings are still debatable whether it's real or not. We would definitely be interested to look at whether SOCS3 expression changes during aging or chronic stress in chondrocytes. It's something that we definitely want to follow up.
Just to add to that, I think most of the studies of negative feedback regulation have been via SOCS have been very short term. So I guess we've been wondering if that could be distorted when the cell is under stress. So say the cell is under a chronic inflammatory stress, whether its ability to switch on those negative feedback mechanisms might be compromised, I think is an interesting possibility. There was either a laugh or a question or a suppressor. Oh no, it's not big enough laughing. No.
I thought you told us that you were studying a cohort of those mice for life, where you knocked out SOCS3 and the chondrocytes. Was I asleep when you told us the answer to that? Did you not really describe the ultimate fate of those mice? Pictures you showed of changes, were they from mice that had gone the whole lifespan? And were the changes the same in multiple joints in the body?
Well, that's a really good question, Don. So we looked at the knee joints of those aged mice at the end of their time points to see whether there's exacerbated spontaneous osteoarthritis that happens. What we found was that in male mice, not in female SOCS3 deleted mice, that they were actually protected, which we don't know what could be contributing to this protection in terms of osteoarthritis, and could go back to Anne's question of in the presence of whether there's a presence of soluble IL-6 receptor there, and whether prolonged interleukin-6 signaling through aging could have added to the cartilage extracellular matrix. It's interesting. I don't quite haven't figured that part out yet. That's why it's interesting to look at.
I mean, the mice otherwise were okay, though, weren't they?
Yeah.
There wasn't premature mortality. There was no obvious sort of non-articular phenotype.
No. So in that particular aspect, that's fine.
Okay.
So you talked a lot about the increased expression of enzymes like matrix and alpha-proteinases in your system. But that's only half the story, because these enzymes have to be cleared to be active, and then their activity is usually mocked up by tissue inhibitors of metalloproteinases. So I wondered whether you saw a converse effect on the expression of the TIPs.
That's a good question, and I was asked that at UCSD on my trip as well. I haven't looked at it, but I imagine that the expression would, if there was an increase in expression, it would have been still lower than the contribution of the catabolic enzymes, which would have contributed to that exacerbated cartilage degradation. But to answer your question, no, I haven't looked at the TIMS, but it will be interesting to look at follow-up on these.
Okay, all that remains is to thank Tommy for being